In this video, we will discuss about CAR T and CAR NK cells designing strategies. So we will start with this CAR T cells, and we have discussed about CAR T cells in one of our previous videos where we have discussed different generation of CAR T cells. We will cover those things again, so it will act as a quick revise for those who have gone through the different kind of CAR T cells video. So this is the CAR design that people are targeting, and what it will does is it will has this T cell receptor complex that can interact with the major histocompatibility complex 1 of the tumor cells to see whether the antigens are present on the tumors, or whether it will see the antigens presence on the cells on which it is interacting, and based on that it will activate the intercellular signaling pathways that will leads to the release of the lytic granules that we discussed previously. So in the first generation CAR, we have the transmembrane domain hinge and these CD3 zeta intracellular domains. In the second CAR, we have this intracellular co-stimulatory domain also. So it will activate the granules formations, or lytic granules formations more. The third generations, we will have two intercellular co-stimulatory domains. In the fourth generations, we have this ZEF70 also that will further accelerate the cytotoxic effect against the tumors. And there are some link CAR T cells also. In this case, link CAR T cells, what will happen? They will inhibit the downstream signaling inhibitory pathways, such as is shown in this figure. There's no downstream signaling activations done, and this is usually occurred on the receptors that has the suppressions actions, or we can say a type of immune checkpoint blockade therapy. So immune checkpoint blockade therapy, we cover the antigens. So in this case, we modify the downstream signaling pathway so the intercellular cells does not get suppressed once they interact with the their once they interact with the ligand that will inhibit the activations of the T cells. And in other case, they will change the intercellular signalings from inhibitions to activations. So instead of suppressing the immune response, it will activate the immune response and reduce uh, and this will lead to the enhanced CAR T cells functions, and also reduced off tumor toxicities. And in terms of the solid tumors, the main challenging about the solid tumors is whether they are able to suppress the whether they are able to overcome the immune evasions or immune surveillance, or whether they are able to overcome the immune evasions that has been done by the solid tumors through various reasons. So first categories is enhanced anti-tumor response. So in this case, they will design the receptors in a way that once it interacts with the tumor antigen, it will release several cytokines such as interleukin 12, 18, 7, 15, 21. And in other case, the intercellular signaling pathways is combinations of many intercellular co-stimulatory domain at the same time. These cytokine receptors are also included. So this will further accelerate the lytic granule released. And in order to block the CAR's cell dysfunctions, what it will does is it will inhibit this canonical TGF beta receptors, or modify these canonical TGF beta receptors so that the macrophages or the signaling pathways that leads to suppression of the immune response will not act. And also this usually what will happen, the solid tumors has this PD-L1 ligand that will interact with the PD-L1 receptors to deactivate or suppress the uh, T immune response. So in this case, the PD-1 receptors on the immune cells can be modified so that intercellular signaling does not get activated. And in terms of this enhance anti-tumor response, other things that can be done is the co-culture these NK cells. So this is about NK cells. So co-culture the NK cells along with this the tumor cells such as K562 or this supplemented media IL-2, IL-21, IL-15 that will lead to the increased ex vivo expansion, and that can be used for in vivo functions. The other things can be done is to prime the cells with this interleukin 12, 18, 15 if they cannot co-culture it with K562 cells. In that case also the metabolic fitness or proliferation survivals in the in vivo increased, and how they overcome the antigen heterogeneity. So in order to overcome the antigen heterogeneity, the they will have designs. They have designs the single chain fragment variable in a way that is more commonly expressed in the tumor cells, or they will design two single chain the CAR NK with that can target two different antigens. The example shown here is the CD73 and CD39. So in that case, one of the approach can be that if the tumor cells has both of these antigens, then only it will activate. It will prevent the non-specific actions of the CAR NK cells or the side effects of the CAR NK cells. Or it can be designed in a OR functions, that is that if it is able to detect CD73 or CD39, then also it will activate. It depends on what kind of tumors the patient has, and how one can improve this CAR cell infiltrations and persistence is by expressing this FAP on the cell surface. By expressing this FAP on the cell surface of the NK cells, it can help in the ECM degradations and thereby helps in the NK cells extravasations and infiltrations. So the other things to improve the cancers, improve CAR cell infiltrations and persistence is through not only through this FAP, but they can also leads to the receptors that will get activate to release this heparanase that will also helps in the ECM protein and proteoglycan degradations, and this can be activated by through these several receptors such as CXCR1, CXCR2, IL8R, and it can help in the CAR T cells migrations and persistence, and it can reach to the tumor cells at distant site. The other things that is mentioned here is about overcoming antigen heterogeneity and loss. So this is similar to what we have discussed for CAR NK cells, but I think on the left hand side this is more about CAR T cells. So in the CAR T cells, they can be done by designing this single chain fragment variables that targeting alpha EphA2, and it can interact with this solid tumors, and thereby leads to the release and thereby leads to the release of this interleukin 13 that can interact with the interleukin 13 receptors that are present on the tumor cells, and this will cause the this will be able to increase the effectiveness of the T cells, as it can not only interact with the EphA2, but it can also interact with these interleukin 13 R receptors. So it will have this dual single chain fragment variables, we can say that, and this can also be designed in the OR manner or in the AND manner. So I hope you understood the CAR T and CAR NK cells designing strategies to like for a beginner point of view. You need to understand more about this CAR T and CAR NK cells designing strategies in detail, or from where you can get some of the good papers. You can go through this paper and its cross references. So that's all for this video. If you need to understand more about this topic in detail, as I have mentioned, you can refer to the paper from which this figure has been taken, that is in the descriptions. So thank you for your time.